Malchus and I are going to help you understand a little bit more about Chapter 15. Now, the textbook is going to call this Evolution on a Small Scale. But really, we're diving into the concepts of natural selection and adaptation. The example from Chapter 14 in that green box right at the beginning of the chapter about antibiotic resistance, that's really going to help us to understand a little bit more about natural selection and some of the problems that we've created for ourselves by not fully understanding our actions and the unintended consequences of our actions as humans. So I love this cartoon because I think it's excellent to kind of make fun of or point at this idea that when it comes to adaptations, we're looking for structures mostly based on genes that are going to give an individual some kind of selective advantage in the environment. And if they have a selective advantage, then their fitness will be improved, more likely to pass on the genes for that structure to the next generation. And so we should see that adaptation, that structure, in this case the ice cream cone, being passed from one generation to the next. Now, this doesn't explain everything that we observe in the natural world, but it does explain some of the observations and adaptations that we see. So, for natural selection, it is important that you understand these criteria. The first criteria or underlying assumption that we have for the concept of natural selection is that individuals in a population have genes. Now, there's going to be some variation in those genes. If we look back at our genetics unit, doing all those Punnett squares was really about looking at the variation that exists and then calculating the probability that those specific traits would get passed on to the next generation. This concept of dominant and recessive became important, but we also started to look at a more common situation where genes are not dominant or recessive. They're either present or absent, or we've got a certain variation or we don't have a certain variation, and looking at the probability of those genes getting passed on to the next generation. So arguably, you've already been collecting data. You've already been dealing with natural selection, looking at the possibility, the probability of traits getting passed on to the next generation. Now, there is this criterion for natural selection that kind of sucks. And that criterion says that individuals will produce more offspring than the environment can support. The underlying assumption here is that somebody's going to die. Quite a lot of somebodies are going to die. And it will only be those that have the traits that allow them to survive and reproduce. Those will be the only ones that get passed on from one generation to the next generation. So we're looking for traits that become fixed in the population. They get passed from one generation to the next with the idea that they're giving individuals some kind of selective advantage. Over time, a larger proportion of the population will have those genes. So I want to give you an example that hopefully you're familiar with of natural selection. In our area, we have the American robin. And for the most part, robins all look the same. But sometimes robins can have partial or complete albinism. In other words, they have patches of feathers or individual feathers that look white. So when we look at this next slide, we have the characteristic robin that we all come to expect here in the foreground. And in the background, we have a robin that has quite a bit of feathering, quite a bit of albinism. It is partial albinism. It's not full and complete because we still have some orange on the chest area and we still have some darker feathers in the wing. But we can see, looking at this, that white is actually going to be a big beacon in the environment. It's going to help this bird to stand out in just about every environment it could be in. So here's some more examples of what we see in robins, that partial albinism. The one thing that we need to keep in mind is that robins are prey for a lot of species. They're prey for other bird species. They are also prey for land species, like this cat here, but also things like foxes and just about anything that could grab a hold of a robin. So a big question is, why do almost all of the robins that we see look the same? So when we look at the biology, the individual biology of the robin, it will help us to understand a little bit more as to why all robins, or almost all robins that we see, look the same. So first thing we need to note is that robins have about 3 to 5 eggs every time they make a nest. And they'll make about 3 nests a year. So on the high end, we're looking at a robin that can make 15 possible babies. But the problem is that only about 80% of the robins will die. So out of the total 15 possible offspring that a female robin could have, only about 3 will make it through their first year of life. Most of them will fall victim to being preyed upon, and any coloration that helps them to blend in will make them more likely to survive, and any coloration that helps them to stand out and get preyed upon, well, they're going to be less likely to pass on those genes to the next generation. So if you have some of that white coloration, you are going to be selected against. Your genes are going to get removed from the population. And over time, what we see is this consistency in the phenotype. Almost all robins look exactly the same. Another example of natural selection that we have happening is with antibiotic resistance. So here in the pictures below, we have this moldy orange. And when we have this coloration of mold, what's growing on it is the Penicillium chrysogenum and other species in the Penicillium family. It's a fungus that's going to be producing a chemical that will selectively kill bacterial cells. A chemical that selectively kills bacterial cells is called an antibiotic. So we find that most of the antibiotics that we use were originally produced by living organisms, like this fungi growing on the orange. Now it's really weird to see this fungus on the orange as being this, tension, this incredible dynamic fight happening between fungi and bacteria. But that's what's happening at the microscopic level. Fungi are secreting penicillin, bacteria are getting killed by it. So it's a great way for the fungus to be able to outcompete the bacteria and get all the goodies inside of the orange. So we got a little bit of background as to why the fungus would make this chemical that selectively kills bacteria. We next need to look at the mechanism for how penicillin will kill bacterial cells. So going all the way back to when we talked about osmosis. Remember that penicillin will weaken the cell wall of bacteria, and that when that cell wall is weakened, water is flowing into the cell. The cell is a hypertonic solution, has more solute. All that water is attracted into the cell, and as that water balloon swells and swells and swells, it's eventually going to hit that weak cell wall, weakened by the penicillin antibiotic, and explode and burst. If the inside cell solution in our bacteria is hypertonic, the outside solution is hypotonic. That's why water is flowing into the cell, causing it to expand and burst when it hits that weak cell wall. So that's the mechanism that penicillin is using to kill bacterial cells. Now, one of the things that we have been doing as humans is we have been using antibiotics in our animal feed in order to grow animals in these industrial situations where we have hundreds, even thousands of animals in very small confined areas, basically for industrial production. In order to grow them in these really hyper-stressful environments, they have to have continual doses of antibiotics from the moment they're born until the moment that they're no longer part of this planet. What this does is it puts selective pressure on the bacteria to develop genes, to develop mutations in those genes that allow them to survive in the presence of the antibiotic. And so in all of our animal populations, on almost every farm that is practicing agriculture this way, there's, if not one, there are multiple types of bacteria that can no longer be killed by antibiotics. So this is a huge problem for animals, but then it's also a huge problem for us humans, because we rely on antibiotics. If you read historical texts and historical accounts of veterinarians and doctors in the early 1900s, before we really started using antibiotics, they're always agonizing. They know what's causing the infection. They know that it's bacteria. They know that simple infections will turn gangrenous and require limbs to be cut off, or in the case of horses and large animals like cattle, it requires those animals to be put down. They knew what a death sentence a bacterial infection could be. And for those doctors who lived through the time when we started using antibiotics, to listen to their joy and how exciting and how amazing and wonderful it was to finally be able to treat those diseases. They had antibiotics, they could do it. And yet here we are, less than 100 years later, and we have developed antibiotic resistance in many bacterial populations. So one of the most common types of antibiotic resistance that we see is MRSA. MRSA is methicillin-resistant Staphylococcus aureus. Methicillin is a type of antibiotic, and it's specifically been used to kill Staphylococcus aureus, which is around us in our environment. It's on our desk, it's in the air, it's everywhere around us. This is a common bacterium. That's the little purple dots in the picture here. And the infection looks like that pretty gruesome infection that we see. So what happens is when we use antibiotics, we are putting selective pressure. When we say selective pressure, what we mean is that only the individuals who develop a mutation to resist this antibiotic, to live, even though they're being given this antibiotic, those individuals will survive. And what they'll do is they'll pass on those genes to the next generation. So in the next generation, we'll be able to live, even though we're giving a chemical to try and kill it. They'll pass on those genes to the next generation, and what we end up with is an entire population of Staphylococcus bacteria that will not be killed. They're giving us the biological middle finger, and they're living anyways, and dividing and dividing and dividing. Now, from an individual point of view, if you've got one of these infections can last months, years. We may never find a way to treat the antibiotic-resistant bacteria that are living now in your body. And that's a really ugly place to be. If we can't kill the bacteria, we're hoping your immune system can work well enough, but a lot of times it can't. So this is a hugely scary situation. When we look at the two ways that bacteria are using to pass those genes on to the next generation, we have to go back to our cell division chapter. Remember in cell division, we talked about how bacteria will do binary fission, where one cell makes two genetically identical cells. So if the first cell has the gene to resist the antibiotic, when it goes through binary fission, it will pass that gene on to the next generation. Remember the video that we saw where those bacterial cells can divide every 20 minutes, which means very quickly you can get a population of bacteria where every single cell cannot be killed by the antibiotic. But what about this situation where the individual bacterial cell isn't actually being exposed to the antibiotic? There's no selective pressure to cause it to develop the mutation that then allows it to resist the bacteria. Well, this is where it gets creepy. Bacteria can use conjunction pili to swap genes back and forth. So, I may have never encountered antibiotics in my whole little bacterial life, but if I slide up next to another cell that has been encountering antibiotics and has that mutation, I push on my plasma membrane and I make this conjunction pili. We can swap genes back and forth, and that other bacterial cell can give me the antibiotic resistance gene. So when we look to soil populations of bacteria, when we look to that, what's called the wild type or the wild environment, outside of humans and outside of our agricultural system, what we are finding is that bacteria are doing this. They are swapping genes and they're giving each other the genes for antibiotic resistance. So we may not personally have introduced the antibiotic to the bacterium, but because other cells had seen it and developed the gene, they can pass that gene to other naive cells that have never experienced the bacterium. So this has been an underlying crisis in our use of antibiotics. And to be honest with you, we don't have a whole lot of solutions. There are many, many researchers who are trying to find new mechanisms to kill bacteria, and they've gotten very clever. We do have more solutions than we did, say, 10 years ago, but because of natural selection, because of what we as humans have been doing, exerting that selective pressure, we have been selecting for and pushing our bacterial species into antibiotic resistance. In other words, we've been stopping ourselves from being able to use the exact tool that 100 years ago people were raving about being able to finally use to kill bacterial species. Well, natural selection. It can be kind of, well, it can bite you in the butt if you weren't paying attention And you didn't know that there could be unintended consequences from something like using antibiotics. So this is why it's important that we understand this concept of natural selection, this concept of differential survival, that some individuals will survive and pass on those genes, and that those genes will become more common in the population, and that it's, for the most part, with natural selection, it's going to be in response to environmental external pressure. Now, there are 3 types of natural selection. So here we're looking at a picture of mice. We're going to use them as our generic example of all 3 types of natural selection. So in our population of mice, we have multiple different phenotypes, everything from super, super light to really, really dark, almost black colored mice. And in our population that's not experiencing natural selection whatsoever, it's just a population without any external pressures, what we see is that there are very few individuals who are light colored and very few individuals that are super dark. Almost everybody's kind of an intermediate phenotype. This is what we call a normal distribution. Very few outliers, very few extremes. Now, if this population of mice is experiencing stabilizing selection, what we're going to see is that those extremes, the really super light and the really super dark, they're going to be taken out of the population. They're going to get eaten. They're going to get eliminated. So the only thing that we are going to see is this consistent intermediate phenotype. We're not going to see these really great big extremes. So we look at populations of things like robins, or if we look at things like elephants or giraffes, or even humans, we can see that for many of the phenotypes, many of the traits, we're going to see that stabilizing selection. Not a whole lot of extremes, and almost everybody is the same in the phenotype that they are expressing. So I want to dive in a little bit with the humans and talk about the perfect example of stabilizing selection. So with humans, birth weight is crucial, or used to be really crucial, for human survival. If a baby was born too light, too not heavy enough, then their organs were probably not developed well enough, especially the lungs and the heart. So anything less than about 6 pounds, and the survival rate was fairly low. If a baby was born too heavy, we had a little different situation. If a baby was born too heavy, 10 pounds and above, the mother may not be as likely to survive. And we're one of those species where our baby's fitness is not only dependent on their own ability to survive, but also dependent on the parent's ability to survive. So if the mother didn't make it through childbirth because of all the damage that was done trying to give birth to this very heavy baby, then the baby was less likely to survive. So in humans, we see the stabilizing selection for human birth weight. Right about 6 to 9 pounds is where most babies will be born. Too heavy or too light, less likely to survive, less likely to pass on those genes to the next generation. And we do have modern medicine that has been interfering with this for many years now. And now babies that are born at under a pound, some of those are able to survive. And that's really quite amazing. And because of C-sections, heavier babies are able to be born and the mother is able to survive as well. So we may see, looking back 100 years from now, we may see that human birth weight changes. But up until this time, her human birth weight is an excellent example of stabilizing selection. What if our population is experiencing directional selection? With directional selection, we generally have one extreme phenotype being selected for. So we see that shift. And the example here is with our mice. Our really light-colored mice are getting eaten. They're not being able to pass on their genes to the next generation. And we see that shift, that directional selection, towards the really dark-colored mice. Now, the book uses the example of horses. And I apologize, I don't know why the book uses this example. Yes, our modern horse is an extreme phenotype. But that's because of our selection, right? We selected for bigger horses. We exerted a lot of pressure for that. So I don't think it's a really good example of directional selection. I think what is a good example of directional selection, where we see that extreme phenotype being selected for, is with our insects and with our plants. We use chemicals to kill insects. We are exerting selective pressure on insects for the extreme phenotype to survive those chemicals. And what we see with all of our insects, whether they're flies or mosquitoes or bed bugs, is that the extreme phenotype, being able to withstand every chemical we can throw at them, that's what we have been selecting for. We look at plants. We often try to kill the weeds, and a weed is nothing more than a plant that is not growing. It's growing where we don't want it. Weed is not even a botanical term. It's more of a social construct. So we try to kill the weeds. We use these chemicals called herbicides. We spray them on the plants, and we are exerting selective pressure. We're selecting for the extreme phenotype for the plant to be able to resist our chemicals. And so in about 2 to 5 years, we've built up that resistance in the population. All we see is that extreme phenotype. So then we develop a new chemical. It works for a few generations, but after a few generations, a few years, the weeds develop resistance. We're locked in a pretty ugly battle with our insects, plants, and the chemicals that we use because of directional selection, a type of natural selection. Well, what if our population was experiencing disruptive or diversifying selection? Well, this one's really interesting because that intermediate phenotype, that is the one that's being eliminated from the population. The extremes are the ones that are succeeding. They're passing on their genes to the next generation. So it almost looks like we have a speciation event. In the case of our mice, we get only light-colored mice and only dark-colored mice and nothing in between. Anything in between is going to get killed. And so down in New Mexico and Arizona, with the rock pocket mouse, this has been happening for many years. And part of that's due to the environment. They have these large swaths of volcanic rock that are dark, almost black in color. And then the rest of the desert areas is a light-colored sand. And so if you're an intermediate mouse, you're any shade of brown, you're going to show up on the volcanic rock and you're going to show up in the sandy soil, and all the predators are going to eat you. You're less likely to pass on those genes to the next generation. But if you're really dark mouse and you live on the volcanic rock, you're not going to be seen. You'll pass on your genes. But if you're a light-colored mouse and live on the volcanic rock, well, you'll get eaten right away. You've got a neon sign saying, "Come and eat me." We see the reverse situation in the sand. Really light-colored mice, able to survive, pass on their genes. Almost all light-colored mice live on the light-colored sand. Dark-colored mice, they've got the neon sign, and so they're going to get eaten and less likely to pass on their genes. So disruptive diversifying selection can be very, very interesting in the natural world. It almost looks like speciation. But we've gotten a hold of the rock pocket mouse genes in light and dark-colored mice. We know that they're almost genetically the same. They're very, very, very similar to one another. So we don't have speciation. We just have this environmental pressure for the extreme phenotypes. The intermediates are disappearing from the population. Disruptive diversifying selection is almost the opposite of stabilizing selection. So we look at natural selection. It's much more complicated than just surviving and passing on your genes. We have to take into account the environmental pressure and everything else leading to what we are observing in our individual little organisms. Now, the next concept that we need to talk about, another mechanism of evolution, is genetic drift. Again, it's important to know the criteria with genetic drift. With genetic drift, we have some kind of random event, whether that's a volcano exploding, a tsunami, all of the hurricanes that are pummeling the different islands in the U.S. and the typhoons over in the Pacific Ocean. Those are definitely random events. The tornadoes. A tornado is a hugely random event to the individual organisms in the path of the tornado. Even human activity is a random event. We put in a road. We tear up part of the prairie of the landscape or the forest to build a house. Our activity is a random event from that individual organism, from the species point of view. So with genetic drift, the emphasis really is going to be on a random event. Some individuals of the population die, and when they die, their genes are eliminated, whatever genes those may be. So only those that remain pass on their genes to the next generation. What we see, what we observe, is this abrupt change in the gene frequency in the population. Over time, we're only observing the individuals of those who survived. It really is seeing the ghost of this traumatic event played out over time. Population size then becomes really important. If you have a large population, genetic drift is less likely. And this makes sense. If I've got 100 individuals and my random event, whatever it is, the volcano explodes, the rock slide happens, doesn't matter what it is. If I have 100 individuals, some of those rare genes, those rare alleles, are more likely to survive because I'm more likely to have more individuals survive. But if I have a small population and this random event occurs, that's more likely that the one individual who had the rare gene dies. And then going forward, I never get to see that gene in the population again. I get that abrupt change in gene frequency. So this is why when populations get really small, we start to freak out as scientists, because any disturbance, any problems, any disease that could go through a population, any random event that could happen is more likely to take out the rare genes, the individual with the rare genes, and we don't know what they need to survive, what every species needs to survive. And so we don't know if they maybe have that magic gene that allows them to survive as the environment changes. So we get small populations, a decrease in genetic diversity, the species is more likely to go extinct. And I think scientists want to call this a law, but being scientists, we get really, really picky about what we call a law. It has to be omnipotent and apply everywhere. We're not certain that this applies everywhere, but just about every time we've looked, decrease the population size, decrease genetic diversity, extinction is bound to happen. So let's look at this generic picture of genetic drift. At the top, we have a population of frogs. And in our population of frogs, we got green and brown. Some kind of random event happens, kills all our brown frogs. From that point forward, for hundreds of years, thousands of years, all we have is the green phenotype in our population. Our abrupt change in gene frequency has occurred. Now let's look at some real-world examples that are a little bit closer to home. The first type of genetic drift that we're going to see close to home is the bottleneck effect. Now, with the bottleneck effect, we have a population that originally had a lot of genetic diversity, like our bottle with all the little colored beads. Some random event eliminates most of the individuals. The few that survive are the only ones that perpetuate the population, so we see that decrease in gene variation, decrease in genetic diversity. So here in Illinois, we have the prairie chicken, and these are adorable little birds. The prairie chicken had a huge range in our state. We are the prairie state for a reason. We used to have a lot of prairie. Because of agriculture, we had shrunk their available prairie down to such a small location in our state. We really only had a few large populations of prairie chickens. So, human agriculture. Our activity was the random event that decreased the population of prairie chickens. So what researchers did was they went and looked at the population of prairie chickens here in Illinois, compared them to the population in Kansas, Nebraska, Minnesota, and what's not shown up here is Wisconsin. And when they looked at the population size, they looked at the number of alleles per locus. In other words, the number of genes per location on the chromosome. And what they discovered is that in all of the states where there had not been a bottleneck effect in the prairie chicken population, they had about 5 to 6 genes per location. Here in Illinois, our prairie chickens had less than 4 genes per location. So who cares? What does that mean? Like, what does that do to the phenotype? So we could count the genes per location. What did that mean for the population? Well, what that meant was that in the populations where the prairie chickens did not have a bottleneck effect, where they had almost 6 genes per location, they were hatching most of their eggs About 85 to almost 100% of their eggs were hatching. In the Illinois population, less than 50% of the eggs were hatching. Now, everybody wants to eat a prairie chicken, except for bobcats. They've studied bobcat stomachs, and they found bobcats really prefer rodents. But hawks and foxes and coyotes, they love to eat prairie chickens, and especially the chicks. They're easy to catch. So if you are a prairie chicken, you need almost all of your eggs to hatch because very few are going to make it to maturity. So if less than 50% of the eggs were hatching, the concern here in Illinois was that our population was going to go extinct. And this was a very real concern. And what researchers had to do is they had to go grab chickens from Wisconsin and Minnesota and these other populations where they had more genetic diversity, bring them to Illinois, hope that they would mate with our Illinois chickens in order to keep the population here in Illinois from going extinct. My understanding is that they are still having to go to the other populations and get prairie chickens and get them to interbreed here in Illinois. We still have not been able to keep that population stable over time. Our second type of genetic drift is the founder effect. And the founder effect is when we have about two, for sake of this class, we'll say 2 breeding individuals go to a new environment. When they go to that new place, that new environment, the new niche, they breed, they make babies. Every offspring in that location can be genetically traced back to the original 2 founding members. And you can imagine that this means there's a lot of genetic similarity between the individuals, and you get a lot of inbreeding, things that make us kind of squeamish and a bit uncomfortable, father-daughter, brother-mother types of matings. But in the wild, if you have nobody else to mate with and the hormones are there, well, you're going to make it happen, regardless of how genetically similar you are. So where we see this happen is on the Isle Royale. And if you're not familiar with it, here we're looking at a picture of Lake Superior. And that great big island in Lake Superior is, that's the Isle Royale. It's about 50 miles long and about 9 to 10 miles wide at its widest point. It is a national park. And if you're looking at this picture and you notice it's a little bit closer to one landmass than the other, you're correct. It's about 13 miles away from Canada. And from time to time, an ice bridge forms. In the 1940s, an ice bridge formed between Canada and the island, and 2 wolves came over. Those 2 wolves then grew the population of wolves on the island to about 50 individuals, and they did this from the 1940s until the 1980s. And so there was a tremendous amount of genetic similarity because all of those wolves, after all of those generations, were descended from the 2 original wolves. The founder effect didn't seem to be affecting the population. Like, they seemed to be pretty healthy. There were 4 or 5 packs at different times. Everybody seemed to be getting enough moose. Then, in the 1980s, the population of wolves experienced a bottleneck effect. The canine parvovirus was moving across the United States mainland and just decimating the canidid family, the coyotes, the wolves on the mainland, the domestic dogs, the foxes. Canine parvovirus is awful. It attacks the cells of the digestive system, and so the dogs ultimately die of, wolves, animals, they die of starvation and dehydration because they vomit and poop themselves to death, and it's really, 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 really painful and awful and debilitating. It's very low chance that an animal can survive once they've contracted the virus. So the island up there in the middle of Lake Superior is really hard to get to, and the National Park Service realized they had some control. They were like, wait a minute, we got this population of wolves. We can just stop people from bringing their dogs to the island. We won't get the virus. We have a better chance of making sure the virus doesn't get to our wolves. But this woman brought her little dog with her and didn't know at the time that it had the virus and was there for a couple of days. And that's all it took. That virus swept through the population. It decimated the population. So we have a bottleneck, a random effect. Woman brought her Yorkie Terrier with the canine parvovirus to the island. Knocks that population down. From the 1980s through the early 2000s, the population started to regrow. But those remaining individuals from the bottleneck effect now had so much genetic similarity. So we had the founder effect, just 2 individuals, and then we shrink the population again with the bottleneck effect. We have these double whammies on the wolf population. And what they noticed is that the wolf population at the island started to have these malformed vertebrae. It didn't seem to be affecting their health. Obviously, we had a wolf, one of the wolves from the original population just died this last winter, the last wolf from that population, and he was 11 years old. So that's a pretty long time for a wild wolf to be able to live. But the decrease in genetic variation was showing up in the morphology, in their physical characteristics. Now, what had been happening to the population since 2008, when they had 22 wolves, is that it had been shrinking and shrinking and shrinking. 2 of the wolf packs got in a big fight. They killed a bunch of their members. And then the last remaining male and female on the island tried to mate, but she would get really crabby and fight him off. And they were having some tension in their relationship. And that was because she was his daughter, sister, or something. Like, it was really, really, they were too closely related. And she was fighting him off, I think, because she was sensing that. Like, whoa, but I want to, but whoa, but what? I mean, it was rough to watch the videos and see the photos as they were doing aerials, monitoring the population. And so what they had to do was they had to go and trap wolves from the mainland, from Canada and Minnesota and Wisconsin, and bring them to the island to reintroduce. Was the decision they decided to make, to try and have a wolf population on the island. They weren't dying because of inbreeding. They were dying because the the moose population was having a bit of a crash as well. And so they're hoping with the reintroduction of wolves, and the moose population in the last 3 years has really kind of exploded. They're hoping that they can stabilize the island again and stabilize this relationship between wolves and moose. But because they got wolves from so many different locations, so many different packs, they were being really intentional, right? They're like, if we're going to do this repopulation exercise, we're going to make sure that we have a lot of genetic diversity. The original wolves on the island were there by happenstance because of a founder effect. But now that we're doing this intentionally, we can have more control over the genetic diversity. So I think it'll be really interesting to see going forward from 2019, when they started the reintroduction project forward, what happens on the island. I don't expect we'll ever see wolves with this kind of vertebrae again. I think that was a result of the inbreeding and the incredible genetic similarities. So this brings me to this point that I've been kind of hitting on throughout this whole lecture. Genetic diversity. Genetic diversity is what gives species the flexibility to adapt in a changing environment. Now, how can you reintroduce diversity? Well, the first way you can do it is through having mutations. But remember, the mutation rate is going to be really low. Generally, we're only going to get 1 mutation in every 10,000 to 100,000 cell divisions. So that means that mutation rates are actually really low for almost all species and almost all populations, even in the face of extreme selective pressure. Another way to get genetic diversity is through gene flow. And with gene flow, what we're really doing is we're taking breeding individuals and bringing them to an isolated population. We're saying, you breeding individuals, you've got interesting genes. Go mate with them. Mix it up. So then their babies will have the genetic diversity. And we're hoping with that genetic diversity that we can then give strength to the population. So with natural selection and genetic drift, we have 2 main mechanisms of evolution. And when we continually look at new species, we find that they fall into one of these 2 categories. We're either making observations because natural selection has been happening, or the observations that we're seeing are because of genetic drift. And the fun part is trying to figure out which one, and then based on which one, what are those subtle details? If it's natural selection, is this population showing a stabilizing, directional, or diversifying disruptive selection? If it's genetic drift, are we observing the result of a founder effect or bottleneck effect? Or potentially, like in the case of the Iowa Royale wolves, are we observing both? As always, if you have any questions, please email me.